So I was thinking we could just go over the whole practice exam.
Yes.
I question marked like 10 of them, so for the first question, I put D. That's what I thought it was. And then
If it's not too much to ask, would you guys be able to read off the questions? I'm at work right now, so I don't have
oh yeah, of course.
the thing in front of me, but yeah, thanks.
So for number one, it's what do you think would happen to the amino acid sequence coded by the DNA below if the DNA sequence were shortened by removing the highlighted nucleotide? And it highlights just a single one. And so I said it would be different after deletion.
Maybe is someone able to maybe share their screen of like the PDF of the practice exam? Maybe that would help.
I can. Does anyone have it pulled up right now?
Yeah, but you have to give. Oh wait, actually no, we can. Yeah, I'll share it.
Okay.
Oh no, you have to make me host or enable it.
Okay, try that.
Okay. Here. Let's see. I'll move that. Okay, that should work. Okay, so this is the first question we're talking about. For whoever is at work and isn't able to see it, can you see it now? Oh, he actually left, it looks like.
All right.
Yeah, I mean, I think it is D. I don't think it's not like it's affecting a starting sequence or
Yeah.
anything else.
For number two, I wasn't quite sure how to do that one.
Let me look at it.
I have like an idea. Maybe you just check the what's it called? The codon table. But if you don't know where it is, then I'm not quite sure.
So we went over this in our discussion today. I don't think I'll do a super good job relaying that, but or relaying like the explanation. But because it says like an early stop in translation, you have to translate it into mRNA, and then you can look at the codon table. Does that make sense?
Yeah.
So okay, so you have to translate them, so it would be 3', and then you would have U, G, G, C, G, U. And then you just do the same for like the bottom one. And then what it's asking, if the difference would cause an early stop, but there isn't a stop codon. So no.
Yeah,
there is not a stop codon. But I think the first step is recognizing that you have to translate it into mRNA, because mRNA is what you use to look at the codon table.
Mm-hmm.
I hope that makes sense.
Yeah. All right, and then for number three, I think I did this wrong because I didn't look at the 3' and 5', but for the blank that's in the question, I put AAG, which is codes for LYS, but I think I have to switch it. What did you guys do on that one?
I haven't done the practice exam yet. This is my first time looking at it.
I may have done that wrong because I get like I'm still getting so confused with like where the 3' and 5' end goes. But
Yeah.
I think the first step is to look at you know that we're given a template strand and we need it in mRNA.
Yeah.
So I think what it ends up being is okay. Which way do you read MR? Yeah, so you read mRNA 3' to 5', right? Okay, so to translate this, it would end up being 5'. What is it? UUG 3', but then you have to read it from the 3' to 5', so it end up being GUU. I might be totally doing this wrong, but that's just what my thought process was. So I actually didn't write down the codon, so let me check the codon table really quick.
So you're saying it would be GUU?
I think so, but I'm like, I wouldn't take my word for anything because I'm really not sure about any of this. That was just my thinking.
What did the rest of you guys put down or think about it?
I haven't done that one yet, so I didn't get it.
Does anyone else have any ideas on how you would like start it? Or like what you would do on an exam if you saw this? And maybe we can all try and work together to figure it out.
I'm trying to find in my notes if it's
I think it might be helpful to look at the discussion sheet when we had to do it. I think it was in something like with the central dogma, maybe. You know, we had to fill it that it was like a puzzle. That
oh yeah.
table.
There's a question like that on the faculty.
Yeah, there is. Yeah, but okay, so it's class 14 worksheet. It has it's like this table. I don't know if you can see that.
Yeah.
But I think that would be helpful because it's, you know, we have the correct answers for that if you did it in discussion. So the template strand in this example is going 3' to 5'. And then the mRNA is 5' to 3'.
Yeah, I think you're right. I think it would be GUU.
Okay.
That definitely makes sense to me.
Are we still on number 3? Sorry, I was setting up my screen.
Yeah, we're on number 3.
Okay.
And then GUU would code for VAL. And then since we know how to do, or since we at least think we know how to do that first one, so we can try A, B, and C.
So we did decide that it was GUU or UGG or UUG.
I think it's GUU because the 5' has to go on the other side.
So, okay. Okay, thank you.
Yeah. And then on the codon table, GUU goes to BAL. And then for letter A, the thing would be AAA. So I think the answer would be UUU, right? And that codes for PHE. If I did that right. And then for letter B, I'm saying it's GAU, and that codes for ASP.
For this one, aren't we supposed to be putting down what kind of mutation it was?
Yeah.
Oh, shoot. Yeah, you're right.
And for A, wouldn't it be AAA?
I'm kind of confused on this one, honestly,
Okay.
because we
Me
have
too.
the DNA template strand, but then we have to change it.
Is there a certain direction you read mRNA? Like, or does it change based off the information?
Hold on, I'll check it. Oh. 5 point. So we translate it from DNA to RNA and then we flip it.
Okay, so then the first blank in the actual question would be GAA. And that codes for GLU.
Yeah.
Yes. Okay. See, that makes
That
more
makes a
sense.
lot more sense. I don't know why I was so caught up on that one.
Yeah.
Oh, so it's GAA for like the blank?
Yeah.
Okay.
And then I don't know the names of what it codes to, but the first three letters
I don't know
is.
if I'm right, but I got a different answer.
How did you do it?
So the template strand is 3' TTC, 5'. So it would be just anti-parallel.
So did you put CTT for the blank and code that?
No, I put AAG.
Well,
So that would be arginine.
You need to flip it because mRNA is read 5' to 3'.
Yeah, it has to be read the opposite direction. Does that make sense?
Yeah, that does.
Okay. All right, and then honestly, I am not good at the mutation part of this.
I think I got this one. So we don't even have to, or as far as I'm concerned, you don't have to change anything. Like you just look at, we're given the first thing, which is 3' TTC.
Mm-hmm.
And then, so if we look at a nonsense mutation is when it changes to a stop codon. So if you look, if you do end up translating all of these, it would be AAA, and then it would be UAC, G, oh my God, UAG, and then UTG.
You have to still flip it because they're all the DNA template strands. So they're 3' to 5', and we need 5' to 3'.
Yeah, so B and C, it would be B is GAU, right?
Yeah.
Yeah.
And then C is GGU.
Yeah.
But then
GGA.
we don't.
I think they're all missense mutations. Because none of them change to a stop codon, and they could be two. I don't think they are though, because it's not like they're the same sequence.
Yeah. Okay, so for
I
the
think for the first one, I think it would be arginine because you would only flip it because you're just trying to base pair it. So if you look on like the table, I think on like page 5 where you have to fill in the RNA stuff. So it would be AAG because you're just trying to find the anti-parallel and the complementary base. And so I kind of use that type of logic to solve the second
because
part.
the given one is the DNA template strand, and it's asking us to use the mRNA. So we have to switch it.
It's not that you have to switch the whole thing. It's just that
It's you
mRNA
just
is
have
always
to,
read 5' to 3'. So you just have to
it's
read it.
made in 5' to 3'. It's read 3 to 5. So if it's made 5 to 3, then that would mean it's anti-parallel and is able to replicate. Does that make sense?
I'm so confused.
So the DNA template strand shows 3' TTC 5',
Mm-hmm.
but we're just trying to make sure that we're able to create like another replica of it, you know, trying to make the non-conservative or the semi-conservative strand.
Mm-hmm.
So we're just trying to find the other part, which is the mRNA. So it would be anti-parallel from 5', and then it would be A matches with T, A matches with T, and then G matches with C,
Mm-hmm.
and then the 3'. We wouldn't need to flip it because then that way
Oh,
we
I see what
wouldn't
you're
be able
saying.
to hydrogen bond. Does that make sense?
Yeah.
So I'm pretty sure that one would be arginine, whatever it is. And then that's the kind of logic that I use to solve the second part. So if I were to just find the complementary base pairs for A, I got AAA, and that one just codes for a different amino acid. And then B would be a stop codon because it translates to UAG. And then for C, I got no change in amino acid sequence since AGG also results in arginine. Does that make sense?
Yeah. So A would be
The number 3, it would be codes for different
the
amino acids.
amino acid to the missense,
Yes,
and then B would be the stop codon to the
the stop codon.
nonsense.
And then C would be the
Would be the
silent
silent.
mutation.
Okay.
Wait, what was your thinking for A? Because you said, wait, what did you say for A? It
So
changes
for
to
A, I just put arginine because the complementary base, the mRNA, would be 5' AAG 3'. And
I'm
we
sorry,
wouldn't necessarily, sorry, for
sorry.
the second part of A.
Yeah, like
Oh, okay.
with the mutations. What did you say for A?
It was a missense because it just codes for a different amino acid. That one would be lysine.
Oh, okay, wait. Are we comparing what we found? Like, are we comparing TTC to TTT? Like the TTC is what was in like the explanation. Are we comparing it to?
No, we're just saying, we're not comparing it. We're kind of just trying to figure out in mRNA, once translated, I guess what kind of, or hold on, let's see.
Because
Yes, so I guess in respect to the TTC.
Yeah,
So it would change to a different amino acid because where that last part changes from a T to a C, the complementary
Yeah.
base is an
Mhm.
A. So it would just be
Mhm.
AAA, resulting in lysine.
Well, AAA and AUG result in the same,
Yes,
they're both
they're the
lysine.
they're the same amino acid,
Yeah.
so there's no change in the sequence.
AAG? I
Yeah,
thought that one was
AAG and
arginine.
AAA are both lysine.
Hold on, AAG. Oh, okay. Oh, hold on. Okay, so then that one would be the missense, and then the AGG would be
Well, I
the
think
silent mutation.
letter A would be the silent mutation.
Okay.
And I think 3 is the missense because it
Yeah.
is just different.
Yeah. Okay. Does everyone feel okay on that question?
So what did we decide? A is AAA, and then B is UAG, and then C is UGG. Is that what it was?
Yeah, and then the mutations, A is a silent mutation, B is a nonsense mutation, and C is the missense mutation.
Okay.
Okay, and then number 4 is just describing the laws. Do we need to go over that?
Yeah, can we please?
Yeah. So the first and second laws, one of them is the independent assortment and then the law of segregation. So the independent assortment is when they just line up randomly in meiosis. And then the segregation law, what I have in my notes is Mendel's law of segregation reflects separation of homologs in anaphase 1 of meiosis. And then independent assortment is random orientation.
Cool. Thank you.
Will you say the not the independent assortment one, the other one again?
The Mendel's law of segregation,
Yeah.
it reflects separation of homologs in anaphase 1 of meiosis.
Okay.
And then we went over number 5 earlier. Do we want to go over that one again? Yeah, okay. I can't remember. Was it you, Sarah, who went over that one? Do you want to go over that one again for the people who weren't here?
Yeah, do you just want me to like say the letters or do y'all need help like understanding the terms?
Just saying the letter works okay for me at least.
Okay, so I did S, and let me know if I'm going too fast. S, G, C, P, R, K, U, L, A, T, B, J, D, E, O, yeah, okay, H, Q, M, N, F, and then I.
What was for the second to last one above I?
F.
F?
I think that would be F2 generation.
Yeah. I thought you said S for a second. I was like,
Oh, yeah, no, you're good.
not sure about that.
Sorry, the last 4 did you say like N as in like phenotype and then M as in wild type?
Let me see here.
Or those sound alike. It's just the last 4 of those. It goes.
I just like erased some of my things, so let me fix this. Okay, so M is a characteristic most commonly found in nature is wild type.
Okay.
And then it's N, which is phenotype.
Okay.
And then F and I.
Cool. Thank you.
Okay, any questions on that one?
Is there H twice?
I only have H on an organism that has 2 identical alleles for a characteristic.
Oh. Then what is it for that has 2 different alleles for a characteristic?
2 different alleles.
Mhm.
2
That would
different. I put B.
Oh,
Yeah,
okay.
it would be heterozygous.
Okay, are we ready to move on to number 6? Okay. So the 2 parents are heterozygous carriers of cystic fibrosis, and then it has what are the chances. For a child affected with cystic fibrosis, I put 1 in 4 because I did the Punnett square and there's only 1 with both of them little a. And then for 2 additional children affected with cystic fibrosis, so it says 2 additional, so it'd be a total of 3 children. So I multiplied 1/4 3 times and got 1/64. And then I'm not sure about letter C. I put 27/64. Is that what you got? Okay. And then you got what?
For B, I don't think it would just be timesing 1/4 by 3. It'd be timesing 1/4 by 1/4 by 1/4.
Yeah, that's what I did.
Oh, okay. Sorry, wording just sounded confused. Sorry.
Yeah, sorry. I'm not sure how to distinguish boy versus girl, so I wasn't sure how to do D or E.
Like for me, there are 50% for a boy and 50% for a girl. So if you like see a question said that will be a boy or will be a girl, just like multiply 1/2 is okay. Like means 50% like of borning boy or girl. But if the question said that it is a boy or it is a girl, means it is a fact, then you don't have to do that.
Okay. So with that being said, letter D would be just half, 2/4, or 1/2.
Like the carriers will be half, right? The chances of carrier, right? But will be a boy means they have half a chance.
Oh, so it'd be 1/4.
Yeah.
Okay. And then their phenotypically normal daughter is a carrier. So being a carrier is 2/4, and then a daughter would make it 1/4 for letter E too.
Being phenotypically normal means
Oh, so
that
there'd be 3.
yeah, so yeah,
3/4.
do one wait.
But if the question says
1/3.
that you don't need to say, like, you don't need to figure out the possibility of it being a boy, then don't you just put, like, the possibility of it being a carrier?
Yeah, so this one says like
For
they're
me-
already daughter.
Yeah.
So it would be
you don't have to take
3/4.
into account.
It
It would be 2/3.
It's 2/3 because it said that like
Oh, yeah, it's not. Oh, a carrier. I didn't read that right.
What did we say B was?
1 in 64.
Okay. Thank you.
Oh, sorry. What was it for letter E? What was the final thing we came up with?
2
2/3.
over 3.
2/3. Why would it be 2 over 3?
Like since we do the square and then we have 4 different like result, right? But only 3 of them are phenotypically normal.
Yeah.
A.
Oh, okay,
A-
okay. I see it now. Thank you. Okay, does anyone else have questions about number 6? Okay, so let's move on to number 7. So I'm not sure how to determine if it's heterozygous or homozygous because they're both gonna look the same.
Yeah, we have to use recessive like bird to determine it is a like homozygous or heterozygous, right? So we have to use the test cross. Means we have to test either big B crossover to small b, or big B little b crossover to little b. Then we have to use the square.
Okay.
And then for the big B little b, big B crossover to little b, we will get 100% all blue.
Yeah.
For the bird, right? But the second one, for the big B little b crossover to little b, we will get the 50% of blue and 50% of white.
Yeah.
So the heterozygous will get 50% blue and 50% white, but the homozygous will get 100% blue. So that's you determine the bird is heterozygous or homozygous. Yeah.
Okay.
Yeah.
Okay, and then for the next couple questions, I'm kind of confused on how to set up the Punnett squares. Because I looked on Google and it gives a big Punnett square with like big A, big B, big A, little b, and like all the different combinations, and then you cross them. So they have 4 in each box. But I'm not sure how to set it up really.
So for that one, you don't need to have like the huge Punnett square. What you can do is just set up a normal one. So just focus on like one letter type at a time. So you would cross big A, little a with big A, little a. Here, let me see if I can, like, this is what I did. Hold on. Let's see. So big A, little a, and that was for the first part. And then this is for the second part. And then when I asked for the phenotype, it just says like the AB phenotype. And so that would mean how many chances of getting a big A in the first part, and then a big B in the second part, and then multiply that. So I got 3/16 for the first one.
Okay. Okay, yeah. And then for big A, little b.
Big A, little b, since there's only 1/4 chance of that happening in the A's, I multiplied that by the 3/4 chance of getting the big B.
Wait, why? Oh, are you doing little a, big B?
Yep, so little a, big, oh, sorry. Okay. I was looking at C.
Oh, you're good. I was like, wait, hold up.
But for B, I also got 3/16 since there's 3 chances of getting big A.
Yeah.
And 1 chance of getting little b.
And then for little b,
I got 1/16 since there's only 1 chance in both.
Yeah. And then I was extra confused on the last couple questions with the whole boy girl thing. And
Oh,
for
yeah.
letter E, since it was,
Yeah, so when it asks for the genotype, it asks like if that's the only thing you can get. So big A, little a in the first Punnett square is half a chance.
mhm,
And it's the same thing for the second one. So it's 1/4.
okay.
And then for F, I got being a male with a B phenotype. So the way that I did this one, hold on, I'm trying to look and see if it makes sense.
Like you did the AB phenotypes on 8, right? The
Yeah,
answer
and then
is
you would
9.
multiply that by half, right?
Yeah, yeah.
Okay, and then so I got 9 out of 32.
Yeah.
And then for the last one, I got 1 out of 64. I don't know if that's what you guys got.
I'm not sure, but I didn't get that. Like, I mean, my answer is not that.
Sorry, what was that?
The last one, my
Mhm.
answer is 1/1024.
Oh,
But can you talk about like how you get it?
yeah. So it says 2 females in a row. So the chance of having a female the first time is 1 out of 2. The second time is also 1 out of 2. So that's 1/4. So it's basically just doing 1/4 times 1/4 times 1/4. So it's also 1 out of 64. Does that make sense?
Yeah, but how about getting your AB phenotype? It's 1/16. So
Hold.
yeah, the first like female who got an AB phenotype will be 1/2 like times 1/16. And the second one will also be 1/2 times 1/16. Then there will be 1/32 times 1/32, right?
Yeah, that makes sense.
Okay. Yeah, I see what you're saying.
Like, just remind that if you see this kind of question, just think it separately. Like
Yeah.
first, like the situation of the first child and the second child, and then just multiply them together.
Yeah, that makes sense. That helps. Thank you.
And then what was letter C again?
3/16.
3/16. Okay. Alrighty.
Wait, sorry. So what was G? 1/1000.
1024.
Okay. Thank you.
All right, and then for these next two, can we still do the 2 separate Punnett squares, or do we have to do one of the big ones?
We just did 2 separate. I mean, we can
Yeah.
always do 2 separate. The
Okay.
only time you do the big one is if like it's just more complicated.
Okay. All right, so
But
what is the chance of big A little b? Wasn't that 3/16?
Yeah, for me, yes.
Yeah.
And then for the next one, let's try this.
For second one, I got 3/8. I don't know if that's what you guys got.
Yeah, me too. I got it.
Okay, so I'm just doing this really quick.
For the like 2 trait, we usually do 2 small square and then multiplies them together, right? But like you have to remember, if we have to do the big one, and then the ratio will be 9 raised to 3 raised to 3 raised to 1. You have to remember that.
So what did you put for the second one?
3/8.
Okay, yeah, that makes sense now. Okay, how many red offspring whenever a dominant allele is present for either or both genes, the red color is produced?
And so this would be the same process, it's just making two Punnett squares.
Mm-hmm.
And it says that the red color
Is
is the
dominant.
dominant allele. So that would mean 3/4 is
Yeah.
what accounts for it. So I got 9/16.
Okay, that makes a lot more sense now. Thank you, guys.
Yeah, the big Punnett square is a mess, so I don't even try.
Yeah, I was like, I Googled it. I'm like, I don't know how to do this. Okay. So for 11, fill in the blanks with A, B, C were not shown. All right, so DNA polymerase for the very last one, I said it was B.
I'm sorry, can you like go to number 10 again?
Oh, yeah.
Yeah,
It
for
was
10,
9
I got
over
9/16.
yeah.
9/16, right? But the question is asking how many red offspring. So do we have to write 9 only? I'm not sure, just ask.
Oh, that's a good question.
I'm not sure.
Yeah,
I
mean,
don't know.
9/16, I'm pretty sure they would still gather as 9 out of the 16th. If we just write 9, we'll probably wonder.
but never mind, we will get MC. You don't have to think about it.
Yeah. These questions are tricky.
Yeah,
Yeah.
I feel like the
I've
last test
probably
was
been to
definitely a
maybe
lot
4
easier.
discussions. I mean, I've probably been to like 4 study halls.
Guys, I'm
And
so scared for tomorrow. Like, I'm terrified of what's going to be on this test.
me too. I didn't do
I don't
too
even
well
want
on
to think
the
about it.
quiz.
Same.
I've been passed one quiz or exam, so
See, I feel like I'm pretty good at biology, and so I've been doing pretty good up until now. And now I'm just like, I don't know what's going on anymore.
Yeah.
Because like I feel like I
feel-
understand it during lectures, and then when I'm trying to fill out this, I'm like, nope.
Yeah, that's how I am too.
I kind of felt like Dr. Fadness had a better way of explaining things. I don't know if it's just me, or I don't know if it's just because this stuff's a little harder.
I'm not sure, but the half, like when Dr. Fennis is teaching, like it's about chemistry, and I'm like, what? I'm taking a biology class. What I'm doing right here, and then but now I think Dr. Fennis is like, I think she teach like well, I think more than Honor. I think.
They have different teaching styles.
either way.
Okay. Yeah.
Whoever's teaching.
Okay, I was kind of confused on number 11. I have 12. I have done some of it, don't worry. But
So for
I'm,
number 11,
yeah.
I was kind of confused as well because we didn't really go over it in our lecture.
Here, I think I'm gonna
But
go.
what helped me is I kind of drew like the cell cycle circle.
You mean for 11, right?
Yeah.
And
it's
not really that hard. Like for A, it's a level of, right? Means doesn't really change at all.
Yeah, and that would be CDK, right?
Yes, definitely. So the second one is A. Because the CDK didn't, like the amount is like fixed. It's only the cycling is like kind of fluctuating. Yeah. And for B is in a S phase. So it's DNA replicating, right? So during DNA replicating, we have the DNA polymers.
Yeah.
So the last one is B.
Okay. And then for RNA polymerase, when I went to a study hall about it, they kind of just said it was not shown since it's not really
yeah, RNA
particular.
Not shown because we don't have any like transcription show here. But
yeah,
like for C is the first one because like the M cycling is kind of a like enzyme who respond to activate the
You only need it up to get to M phase, right?
yeah. So
Okay.
it's C, the first one.
Okay.
Okay. Then for 12, I'm just going to read down the list of what I got and stop me if there's a just like a difference. I put A, B, 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 C, B, A, C, C.
For the
For the
second,
second to last one, yeah, I
Yeah.
got B.
me too.
For the
For the
homologous
homologous
chromosomes.
chromosomes undergo synapsis.
For letter B.
Yeah. I put that one only happens in
Oh,
meiosis.
yeah. Yeah. Okay. Okay, so let's move on to number 13.
Sorry to go back. What was RNA polymers on 11?
That it was not shown.
So just put not shown.
Yeah.
Yeah, that's what I put.
Okay. Cool. Thank you.
I was trying to make sense of number 13, and I feel like each study hall I went to kind of had different answers, and they were kind of just saying that it's up to like, it's the question like this wouldn't be on an exam. They just want to stress the importance of meiosis and when the amount of DNA changes, because where it says 14.6 picograms, that one would be mitosis all the way through, since that one doesn't change ever. After S phase, at least.
Yeah. And then did they say what the first two were?
Not really. I mean, for the 3.7, they kind of just said that's when it would be once they're gametes. So they didn't really list a cell cycle stage. So I guess what I wrote was after telophase 2 of meiosis, since that's when they're like separate, or even anaphase, since that's when they move to separate sides. But
yeah,
it's kind of, I don't know if anyone else has any other thoughts on that.
Yeah, this one really confused me. Okay, should we move on to number 14? Since they said that this one is the number 13 isn't going to be on an exam. All right, well, for 14, I put letter C. Specific stages where further progress of a cell can be halted. Did you guys?
That's what I got too.
Okay. And then for 15, I put A because it's asking the anticodon. So it would, the amino acid would be the opposite of that. So 5' to 3', it would be UAC. And that codes for tyrosine. Did you guys get that too?
I got histidine because when the anticodon is 5' to 3', you like the parallel or the complementary was UAC.
Yeah.
But then and then they had me flip it. So it would be 5' CAU, 3'.
CAU. Okay. That makes sense.
Since on the mRNA codon table, you have to go from 5' to 3'. Does that make sense?
Okay, and then for number 16, the template strand was TAC. The anticodon that pairs with the coding mRNA should be, if it's asking the opposite of the mRNA. I put letter C, but I definitely could be wrong. What did you
That's what I
guys?
got too.
You got C? Okay. All right, and then I sucked at these tables, so I kind of filled in what I could, but I definitely did not do the majority of it for number 17.
I kind of struggled with like when it says which way they're read.
Mm-hmm.
That kind of confused me a little bit. So
So
I did the first chart, but I did not do the second one.
So I think the only thing that changes when it's saying left to right versus right to left would be the polypeptide, and you would just read it from. So for example, on chart B, the tRNA anticodon is GCA. So for the polypeptide, you would read it ACG. I think.
Okay, I see. Yeah, I see
That's
you're
what I
talking about.
did.
And then what I did with the second one, where it says like TRP for the mRNA codon,
Mhm.
that one's supposed to be GGU instead of UGG. Does that make sense? Like right to left, GGU. Otherwise, it wouldn't code for TRP.
Let me look at the table real quick. Yeah. So on the table, it's UGG, so it would be right to left would be UGG, yeah. Okay. So on letter A, it would be UGG.
Yeah.
No?
Is it
Wait.
UGG for the mRNA?
Yeah, for A.
Yeah. Okay.
Okay. Does someone want to go over their grids? Because I'm not gonna lie, I didn't do this one.
Yeah, so the template strand is the top strand. And I kind of gathered that because we're given in the very last little quadrant, the UTGA. Do you see that? And
Yeah.
since in DNA, U is replaced by T, I kind of gathered that that was going to be the coding strand, because if it were to be complementary, then we would have gotten A instead.
Yeah.
And then I'm not sure, I guess, where else you guys want me to talk about. I can go over the whole thing. I don't know if you guys want me to go like left to right or how, or like in columns.
I think left to right would be easy. It's just like going across. And so we could just check what we have.
So for the DNA template strand, I got CGT, ACC, GCA, ACT, and that one would be 3' on the left, 5' to the right. And then for the coding strand, I got 5', GCA, TGG, CGT, TGA. And for the mRNA codon, it's basically the same as the coding strand, except replacing the T's with U's. So 5', GCA, UGG, CGU, UGA, and this is where I got all the polypeptides from. And then the tRNA anticodon, I got CGU, ACC, GCA, ACU. And for the polypeptide, I got the N end on that box. And then alanine, So ALA. And then TRP was given to us. And the other one is arginine, ARG. And the last one is a stop codon. So I just wrote stop.
The ones I did matched up with yours. So
Okay, but don't I guess don't ask me how I did do B. I don't really know.
okay.
For the tRNA anticodon, what did you get for the like the primes?
What question are y'all on? Sorry, I was just working on the practice test.
We're on 17.
On 17 doing the tables.
Could you guys come back quickly to 8? I just want to check what I got.
Yeah.
Perfect. Thank you guys. All of you saints.
8, 8A, B, and C were all 3/16. D was 1/16.
A was 9/16.
Yeah, wouldn't be
Yeah,
9. Okay.
Sorry, I read it wrong. E was 1/4. F
D.
was what?
Wait, wouldn't D be just 1/16 because you have to have both of the
Yeah.
units? Okay, thanks. Okay.
E is 1/16. E is 1/4. F was 9/32. And G was 1/1024.
Okay.
Okay, do we want to move on to 18? For 18A, the ball, I put amino acid. For letter B, I put tRNA. C was mRNA. D was a large subunit of the ribosome. E was polypeptide. And F is translation. And then for letter G, I put that the arrow is going to the left. And then the 3' is on the left, 5' is on the right. So A is the one on the left, the A site is the one on the left, P site is in the middle, and E site was on the right. And then I put the start codon on the right side, reading right to left AUG.
So that's what I got too. And then I put the N end on the top of the polypeptide chain.
Wait, did you put the start codon on the left or the right?
On the right.
Okay.
And then for 19, it's kind of hard to do this over Zoom. But I only got a couple of them. I was kind of confused. So for the 5' cap, I said it was the little blue arrow that's on the middle one. For
I can show you guys mine.
Okay.
That doesn't necessarily mean it'll be right. It's kind of a mess because there are a lot of terms that we have to use.
Yeah.
So the Tata box in the promoter region, I guess it's like synonymous, kind of. So the promoter region I put right here, the Tata box right under it. And then in the study hall, they were kind of saying that like the transcription start site and the terminator were like up to interpretation. It was really anywhere that you wanted to put it.
Mm-hmm.
Well, I guess not where you wanted to put it, but where it makes sense. So I put it right here, like start site.
Yeah, that's what
And
I
then
did.
the little red lines in the bottom, I put
Or
the introns,
ABCD,
so
yeah.
A, B, C, D. And then the exons, I wrote in both of the DNA and mRNA. So you guys probably gathered the first one was DNA, second one's mRNA, and the last one's the polypeptide.
Yeah.
And then it's transcription and then translation. And the 5' untranslated region, I kind of put in this like little black area here.
Yeah, me too.
since it just keeps going. And the same for the 3' untranslated region.
Mm-hmm.
And the splice sites, I kind of just drew in between the exons and introns. I'm not exactly sure like what these given lines are supposed to indicate because
Yeah.
I didn't use them in the study halls that they gave us. Nobody used it. And then the terminator I put like towards the end. And 5' cap is a little blue thing, and the
Yeah.
3' poly A tail.
Was the
And
A tail.
yeah, and then I just put translation start by the 5' cap and translation stop by the 3'.
That's what I
And then
did
the
too.
N end and the C end. So N end on the left and the C end on the right for the polypeptide. I don't know if I might have missed any of them.
That matches what mine is. Does anyone have questions on that one?
Where did you put the exons?
The exons, they're the red parts.
Yeah, so I just
Oh,
wrote the
and then
number
the-
above it.
Okay. And then the splice sites are the little like dashes in the mRNA.
Mhm.
Splice sites, I kind of just drew like a line in between the introns and the entrons
Mhm.
because like that's where it would be spliced, but because the lines that they gave us don't really line up.
Yeah.
Okay.
Okay. We're almost done. 3 more questions. 4, I guess. So for 20 and 21, they're kind of the same. Well, they're similar. I was kind of confused on how to do these ones. Because we know that it's DNA and we have to, I don't know. I'm just kind of confused on these.
I have a hard time with these too. Like the one on the quiz really like screwed me up because there was no arrow.
Yeah.
And I find like that's helpful. But on the top one for the promoter, it would be, so the incoming RNA would be synthesized 5' to 3'.
Mm-hmm.
So the bottom strand would be the template strand. And then you just do the complementary base pairings depending on that. So it would be 5', AUG, UGG, CAC, and 3' is the mRNA.
Yeah, that makes sense. Okay.
And then the similar process for the bottom one.
Mhm.
But for the bottom one, I like found the mRNA because I wrote 3' and then everything else and then 5'. So I like flipped it. I don't really know if that's something that they're going to be super strict about on the exam if they give us something like this.
Mhm. Okay. Are we all good on those two? For 22, I was kind of tossed up between C and D. I opted to go with D, but I'm not quite sure. What did you guys put on 22?
I got D for that one because I feel like for C, the promoter regions in front of each gene, I feel like that kind of doesn't really mean anything.
Yeah, that's why I went with D.
Because it can be like if you look at 20 and 21, it can be indicated either way. Like if it's flipped, it really depends on how you look at it.
Mm-hmm. Yeah. Oh my gosh, my table just fell apart. Okay. For 23, I wasn't completely sure on this, but I put C because I feel like the DNA is going to be the same, but the proteins can change, and that's what kind of helps with the variety.
Yeah, that's what I got too. Because all
Okay.
DNA is, the sequences are going to be
Yeah.
similar, or the same.
Hmm.
So it rules out 3 of them already.
Yeah.
And then I settled on that one because if there's different proteins, I kind of thought of it as there are different transcription factors. That
Yeah.
makes sense.
Mhm.
Kind of like that same question on the practice quiz.
Yeah. Alrighty. Does anyone have any other questions while we're all here?
Yes,
I guess good luck, everybody.
I got the 17
Yeah.
for the polypeptide. The A is N and then the B is C, right?
For which one?
Yep.
Yeah, okay,
Okay.
thank you.
Yeah. So I kind of just, for that first column where it asks you to write like the polarity, I basically just flipped it. I did the opposite on the second one.
Alrighty. Well, I'm gonna stop the recording now.